Hi class, this is a quick recording regarding the material in Unit 1. And Unit 1 is related to biosafety and aseptic technique, and you are supposed to complete 2 labs or simulation related to this. So let me get started with biological safety levels, or BSL. So laboratories, microbiology laboratories have levels of biosafety. And these, I recommend that you read more in detail in the theory document of the Labster simulation, because it's going to go more in details of what those levels entails. But basically there are 4 levels, BSL 1 to 4, and the higher the level, that means that the more dangerous microbes are handled there, which requires both more complex instrumentation and just the setup of the lab, and also more stringent practices. So BSL 1 is basically a lab that handles minimally hazardous bacteria. BSL 2 would be the most common setting in microbiology labs. I like teaching labs at universities, and most clinical labs are BSL 2 level, which means that a number of precautions are taken. There is an autoclave to sterilize the media and the materials before and after the manipulation. BSL 3 means that those microbes can spread airborne, and usually it requires that there is a negative airflow in the lab. And in the most common setting, that would be, for example, labs that handle tuberculosis, Mycobacterium tuberculosis, or TB samples, because this is a bacterium that can spread airborne. And this is also the topic of the Labster simulation, in which you are handling an anthrax sample, Bacillus anthracis. We are going to learn in the course that bacilli, so the group of gram-positive bacteria called like bacilli, can form spores, and spores are like this very dry powder, which can obviously spread through the air and can be inhaled. And BSL-4 are, and I'm going to show you this picture here, so these are agents that are extremely dangerous. The minimal exposure to the skin or respiratory pathways can cause infection. An example would be Ebola, and here are a number of steps that should be taken. And during the Ebola, you may remember a few years ago, there was a few Ebola cases in the United States that nurses and healthcare personnel had to take extra training to learn how to handle these samples. So I recommend that when you do the biosafety simulation, really follow the steps that are being told, because otherwise you may die, or your co-workers may die in the simulation, and you will see a number of these very repetitive, very scripted steps, but the reason is obviously to prevent accidents or contamination. Okay, so let's move on now to aseptic technique. This is a critical principle of microbiology, and I would say it's not only for microbiology. Anybody working in healthcare settings, especially before a microbiology lab, the samples have to be taken from patients, and it's critical that personnel knows how to take those samples and how to handle those samples to minimize contamination. And I do know for a fact that blood cultures, which are critical to diagnose, for example, sepsis, are notoriously prone to contamination, because when the samples are taken, they are very often contaminated with microbes from the skin of the patient. So again, it's a set of principles practices. They reduce the potential of unwanted microbes in the culture, but also it protects yourself. Inoculation is the step of introducing the sample, and that sample is called the inoculum. So let's say you take a sample of urine or blood or saliva, etc., nasal swab from a patient. That's going to be your inoculum, and then you are going to introduce it in a culture medium. And the sterile technique Again, has this double purpose of protecting your culture, so you're only going to get the growth that you want, plus protecting yourself. And the way you do it is, one, the materials you work with are going to be sterile, so instruments and culture media are going to be sterilized, and also the practices you follow are going to minimize the risk of contamination. So when you do the aseptic technique labs or simulation, you are going to see emphasis of some of these practices. Like it tells you, touch that blue button and check what happened. And if you did something wrong, it will take you back and you have to repeat it. So some of it is just keeping your area where you work clean. So you have to wipe it down, for example. In the biosafety simulation, in the aseptic technique simulation and in real life, often you'll be working close to a flame, a Bunsen burner. And the reason is, one, because you will be sterilizing some of the tools and materials with the flame. And the second is because around the flame, because of the heat, there is going to be a cleaner, sterile zone. So in the simulation, tell you work close to the flame, and this is the reason for. Anything that can be flamed, and this is usually metal loops and needles, and even spreaders, which are glass, they can be flamed briefly, should be flamed. And the way you do it is you pass it through the flame and then you wait until it cools down. Many clinical labs use disposable materials. They are more expensive, but at the same time they reduce the time of application. And these disposable tools are plastic, so clearly they cannot be sterilized. But what it means is that you also have to handle them with care, because if they get contaminated, you have no way to rectify the situation. Another general principle is whenever you open something, and that could be a tube or a flask, you want to flame the opening of that tube or flask both before and after, and that is to prevent, if there is going to be contamination, is going to be to go through the opening of that tube or flask. So you want to flame it briefly. And in general, just, anything that you touch your culture with, it can be a loop, especially you don't want to touch anything else during the process. And if you do, you have to repeat the serialization and work carefully and fast. So these are a little bit like playing an instrument. You get used to it the more you do it. Okay, so I'm going to go through the steps of transferring bacteria from one culture to another, and this makes much more sense if you're actually watching a video, but follow me. I'm going to play air guitar here. So you're going to have your loop in your dominant hand, and your other hand is going to be handling the tubes or flask or whatever you are using. So you take your loop and you flame it. So you pass it through the flame until it becomes red hot. It glows orange. And from that point and on, you are going to keep your loop in the air close to the flame. You are not going to put it down because that would contaminate it again. You have to wait for the loop to cool, because if you put your red hot loop into your bacterial culture, that's going to burn your bacteria and kill them, so they will not grow. So you have to let it cool. Then you take the tube with the bacteria, and there is a way to do this, and so I'm holding the tube, and then with my pinky finger, I'm still holding the loop here. Actually, let me hold a pen here. So I'm holding the loop here, and then with this little pinky finger, I'm holding the cap. Then I flame the tube. All right. So now I'm still holding the cap. So now I go in the tube. So now I have the bacteria in the loop, and then I flame my tube again, and I cap. So this is the original tube, and I put it down. This loop has the bacteria, so I'm holding it in the air carefully. Now I take my new tube, and actually I think I have to change the slide. Okay, so I take my new tube, take the cap, run it through the flame. Remember, this is the loop with my culture. Inoculate it in here, flame the tube again, cap it, and put it back. This loop still has the bacteria, so now I'm going to flame it again, burn them all, and now I can put my loop down. Okay, so again, watch the video. It's going to make much more sense. And now you can start your culture. Now, what I just showed was about a tube, which you could be inoculating, let's say, a broth or a deep. If it was a plate, I can do additional things to it, and this is something we are going to see in further units. So you can do what is called a spread plate or a streak plate. If it's a plate, it's a Petri dish with a lid, you are to incubate it in the incubator upside down. Now, 37 degrees is kind of 35, 37 degrees is the range of the human body. So this is specifically for microbes that are isolated from the human body. So mainly clinical culture. You can set the incubator other temperatures depending on the temperature that your bacteria like, and we'll get there when we get to microbial growth. So again, why do you have it upside down? Because otherwise the moisture remains, so you're going to get drops of liquid inside the lid, and then they can drop on the culture, contaminating it. So that's why we always have the plate upside down. So that was the summary of the aseptic technique. Watch the videos, do the simulation. It's going to make much more sense, and be sure that you are clear with the BSL levels and what kind of microbes used for which, and also all these steps in doing the aseptic technique. Thank you.